A healthy leaf operates as a high-volume solar factory, swaying in the sun while converting light into the energy that fuels the plant. Aphids disrupt this production. They colonize the surface, tracking along the primary veins to find a path into the interior. The attacker uses a stylet, a specialized biological needle. It plunges through the clear epidermal layer, hunting for the nutrient-rich sap within. Because plants are rooted in place, they cannot physically flee or dodge this kind of mechanical breach. While the leaf looks static, it is actually primed to respond. The moment that stylet breaks the surface, a sequence of structural and chemical defenses begins. The first obstacle is a terrain of trichomes. These surface hairs force the aphids to navigate a dense thicket, exhausting them before they can even begin to feed. Glandular trichomes provide a more active trap. They release sticky droplets that entangle and immobilize the insects on contact. The leaf surface is also coated in a cuticular wax layer. This creates a hydrophobic, low-friction environment where aphids struggle to maintain their grip. This specialized architecture turns the leaf surface into a hostile landscape, designed to neutralize threats before they reach the internal tissue. If an aphid survives the surface, its stylet must still navigate between the plant's internal cells. The surrounding cells detect the mechanical pressure of the stylet and begin to reinforce their outer walls. The plant deposits lignin, thick, dark lines reinforcing the cell walls to block the probe. A sticky pectin matrix floods the intercellular gaps, acting as biological cement that forces the stylet to bend. The plant forces the aphid to expend significant energy for every micrometer of depth, turning a simple puncture into a difficult excavation. When a stylet finally reaches the phloem, it taps into the tubes carrying the plant's sugar supply. The pressure drop triggers an immediate alarm. A surge of calcium ions, visible here as a glowing wave, rushes through the breached tube. This calcium signal activates proteins called ferrosomes, expanding instantly to choke off sap flow. To ensure a permanent seal, the plant deposits callose over the sieve plate, creating a solid plug that isolates the damage. By sacrificing a localized part of its vascular system, the plant prevents the aphid from draining the entire organism. The internal blockades starve the current attackers, but the larger colony on the surface still poses a significant threat. The leaf now deploys a chemical defense. It synthesizes volatile organic compounds and releases them into the air, as shown by these diffusing molecules. Now, these airborne signals are highly specific. They act as a homing beacon for predators that specifically feed on aphids. A ladybird beetle, lured by these specific chemical cues, arrives on the damaged leaf and immediately begins to feed. The predator rapidly clears the remaining colony, allowing the leaf's ecosystem to return to a stable state. Ultimately, a leaf survives through coordinated action. It manages its defense from the microscopic level of a single cell up to the ecological scale of airborne warfare.